Good afternoon. This is the first installment of your online video lectures. As you may be aware, we are transitioning to online lectures moving forward. I hope that everybody is doing well, first and foremost, either physically or mentally. I know there's a lot of changes. I hope that we can continue with the same cadence and schedule that we had outlined at the beginning of the semester through the syllabus. I know a lot of you are taking this course as a goal towards a future career, and we can see that now more than ever we need people within the healthcare industry, people that are competent, and people that are well versed in anatomy and physiology. On that tone, let's get started with chapter 19, blood. The objectives for this chapter is to put the cardiovascular system into this high-level perspective, describe the components and functions of blood, the characteristics of red blood cells, their components such as hemoglobin, the production, maturation of red blood cells. Also explain the importance of blood typing, especially whenever it comes to incompatibility issues. We're going to talk about white blood cells, their function, and we're going to talk about platelets and their role within clotting. So let's start with the cardiovascular system. This is the function of the cardiovascular system. Transport of gases, waste products, nutrients, and all of these are essential components for the proper functioning of cells. So basically, what do cells need? A cell needs nutrients. A cell needs to get rid of metabolic waste. A cell needs to communicate with other cells. Otherwise, it'll be in isolation, in quarantine, if you will. A cell also needs the appropriate environment for these metabolic functions to occur adequately. So it needs the appropriate temperature, it needs the appropriate pH, the appropriate osmotic balance. All of these homeostatic inputs are buffered and are regulated by the blood. Whenever we talk about blood, blood is a type of fluid connective tissue. We mentioned the definition of connective tissue is an specialized cell type with a matrix around it. In this case, the matrix is plasma, and the specialized cell types we'll see are red blood cells and white blood cells and platelets. Let's talk about the function of blood. We talked about function of blood in this high-level context. Specifically, anything that the cell needs, gases, oxygen, CO2, nutrients, hormones, get rid of metabolic wastes, pH, ion composition. Also, blood needs to maintain its own homeostasis. So if homeostasis gets broken by the literal breakage of blood vessels, blood has a way of maintaining that, of returning to homeostasis. White blood cells are in charge of fighting pathogens, and blood is responsible for regulating body temperature. The composition of blood, we have 55% of it being plasma, the inorganic matrix, and the specialized cells being red blood cells, white blood cells, and platelets, which compose about 45% of a whole blood. If you centrifuge blood, fractionated for analytical purposes, you will be able to tell if there's any sort of infection or if there's any sort of anemia just by looking at these percentages and the relationships to them. So let's start with taking a closer look at plasma. Plasma itself, here is the range of concentrations of plasma. We have plasma proteins representing 7% of plasma, other solutes 1%, which includes electrolytes, organic nutrients, organic waste. This is urea. And mostly it's water. 92% of blood is water. That's why it's so fluid. That's why whenever you get stabbed, you start leaking. So the plasma proteins, we have albumins, globulins, and fibrinogen. We're going to look at them in depth in a little bit. The form elements of blood include white blood cells, platelets, and red blood cells. Most of the form elements are going to be red blood cells. That is the main function of blood, a transportation of gases. We gave an example in class. You can go without food for months, in theory. You can go without water for days. You can go, but you cannot go without oxygen for more than several minutes. Just hold your breath, see how long you can do without it. Blood is the main conduit for this exchange of gases to provide oxygen. Most of the form elements of the blood are going to be red blood cells, but we also have white blood cells and platelets. White blood cells are going to be neutrophils, eosinophils, and basophils. These are known as granulocytes due to these granules that are all around it. And we have agranulocytes, monocytes, which are also known as macrophages. Once they leave the blood vessel and infiltrate tissues, because infections happen everywhere, and these macrophages need to get there. And we also have lymphocytes, which we have three categories: B lymphocytes, T lymphocytes, and natural killer cells. We'll look at them in depth in a minute. The B lymphocytes are the ones responsible for creating antibodies. Usually we have memory cells that are able to, whenever we get exposed to a particular new antigen or a novel virus, and we develop the antibodies for it, we have memory cells that retain that memory, and whenever we get exposed to it again, and that memory cell knows how to make that antibody, and it just reproduces really fast, and we have this really concerted and strong response towards that antibody. That is the basis of vaccination. They expose us to an antigen, and we are able to create a response. And that way, the next time that we're exposed to that antigen, we have the memory cells that are able to divide and attack that particular antigen, virus, bacteria, any foreign invader. These form elements, they arise from these hemocytoblasts. Hemocytoblasts that differentiate into myeloid or lymphoid stem cells, and from these hemocytoblasts, all form elements of the blood arise. And from lymphoid stem cells, you have lymphocytes arising, and from myeloid stem cells, you have all other white blood cells and red blood cells and platelets arising. These are properties of blood. Their temperature is at 100.4 degrees, and that's higher than body temperature. Blood is responsible for maintaining the temperature homeostasis within us due to the fact that it's mostly water, and water has a high heat capacity, so it can store a lot of heat. Whenever we go for a run and we are we're overheating. Our skin serves as a radiator of heat. That's why your cheeks get rosy, because blood goes to the surface in order to exchange with heat with the exterior environment. As that water evaporates, it takes a lot of heat away from you and you cool off. It's a slightly alkaline, at a pH of 7.3, and it's 5.5 times more viscous than water. That's due to the plasma proteins that we alluded to earlier. Those plasma proteins remain within the blood. There's a lot of exchange between blood and the interstitial space that surrounds cells. That is the goal of blood, providing those gases, those nutrients, that heat towards cells. And it does this through this intermediary, this interstitial space. It rarely goes from blood to cell. So let's take a closer look at plasma. Plasma accounts for, we mentioned, 55% of blood volume, 92% water, 7% proteins, 1% solutes. Plasma has a higher concentration of oxygen and dissolved proteins for the reasons we mentioned. Proteins cannot readily leave the blood vessels, and oxygen is in higher concentration to increase. This creates a gradient between the red blood cells and the interstitial space, so that there can be exchange of gases, oxygen in one direction, CO2 in another direction. Plasma proteins, specifically looking at plasma proteins, 90% of them are synthesized in the liver. 60% of them are albumins. Due to their quantity, they are responsible for that viscosity and the osmotic pressure of blood. Osmotic pressure is given by the number of solutes that are within blood, and in this case, there's just more proteins within blood than the interstitial space. The albumins are also responsible for the transport of lipids and hormones. Lipids and hormones that are hydrophobic in nature. We mentioned if you were to to pour oil into your sink, the oil does not readily flow. It sticks to the walls of the sink, and this is because it is hydrophobic and it tries to avoid as much contact with the sink, and it does so by creating these droplets. It doesn't expand because that means more surface area, more contact, and it sticks to the walls for that reason. We don't want that to happen within our blood vessels, and for that purpose, albumins create these capsules that surround triglycerides, that surround lipids, and they help transport them from point A to point B. Globulins, they make up 35% of plasma proteins, and there are the immunoglobulins and transport globulins. Immunoglobulins are your antibodies. They attack foreign pathogens, and whenever there is a novel virus, you might not have the antibodies towards that virus. Transport globulins perform the same role as albumins whenever they transport lipids or hydrophobic hormones such as thyroid stimulating hormone. And they also transport ions, which are highly reactive, and we don't want them to be just reacting anywhere they go. So they have these chaperones, these transport proteins that get them from point A to point B. Fibrinogen, 4% of plasma proteins, they get converted to this insoluble fibrin that creates a network that during the chlorine process creates a barrier that prevents further blood loss. If you remove fibrinogen, you will no longer be able to create these fibrin networks that create these coagulates, And this is beneficial whenever you're talking about clinical outcomes, when you don't want blood to clot, basically. And this blood with removed fibrinogen is known as serum. Erythrocytes account for 99%. We mentioned of foreign elements. Your hematocrit is this centrifugate of blood that allows you to analyze basically the quantity of foreign elements. If there are too many white blood cells, or you might have an infection. If there are too many white blood cell precursors, you might have unregulated cell division. This might be a leukemia. If there is a high level of red blood cells, there might be doping involved, or you might live at high altitudes where you need more red blood cells. Hemoglobin is the protein that gives blood its properties that allow it to exchange large quantities of gases, of oxygen and CO2. So let's look at the structure of red blood cells. The red blood cells are biconcave, as you can see here. Why are they biconcave? Why aren't they circles? One of the benefits of being biconcave is that you increase the surface area. You increase the surface area and allows exchange with between red blood cells and the interstitial fluid and the cells that reside within this interstitial fluid. Cells are blood is circulating constantly. It does so slower within capillaries, just so there is more greater chance for escape, greater chance for exchange. But this exchange happens just based on gradients. The concentrations of the large quantities of hemoglobin, the biconcave shape of the cell, all of these properties allow there to be greater exchange. The shape allows it to create these stacks. Basically, they create these trains of red blood cells called rologs that allow them to traverse the small aperture of capillaries in a more smooth manner. Red blood cells lack organelles, and including nuclei. And for this reason, this is of benefit because it allows them to be packed with hemoglobin to exchange oxygen. However, whenever you lack this protein machinery to build proteins, you are not able to. If there is any insult towards the cell, you're not able to respond. So in that sense, they usually degenerate in 120 days. Here we have a view of red blood cells. This level, you can see that they're about 8 micrometers in length and 3 micrometers in height. Usually you can use within microscopy preparations red blood cells as a rubric, as a ruler to determine what the other structures are in relationship to, because there's blood everywhere. In every histological preparation, you should see blood. Here we have that example of rologs. This is just stacks of red blood cells. They form trains that pass through, that orderly pass through small capillaries. Now we mentioned red blood cells, but we also have blue blood cells, green blood cells, purple blood cells, and that is dependent upon the metal used to bind to oxygen or CO2. This is in the case of red blood cells, you have iron. In case of hemocyanin, you have copper. Green blood and purple blood have iron as well. However, this iron is not just freely flowing. This is bound to this inorganic moiety. In the case of red blood cells, we have a heme group. And in case of the other types of blood cells, you also have this inorganic moiety that gives them these visual characteristics. Red blood cells usually have about 250 million molecules of hemoglobin 250 million proteins of hemoglobin within every red blood cell. Each one has 4 heme groups that can bind to, each one will bind to 1 oxygen. So every red blood cell can carry about 1 billion molecules of oxygen or CO2. This hemoglobin is composed of 2 alpha chains and 2 beta chains. Whenever there is any sort of disruption towards the phenotype of red blood cell, blood would not be able to perform its function. So its function of providing oxygen, of regulating temperature, regulating pH, providing nutrients. So all of those symptoms will be that if you're not able to perform these functions effectively, you will have symptoms that correspond to that. So you're not able to provide oxygen correctly. There will be some sort of anosmia. So this is one example of the sickle cell anemia, in where this is caused by an autosomal recessive single nucleotide polymorphism. Basically, it's an recessive allele, and there's only a mutation in 1 nucleotide. But that mutation allows the change from glutamic acid to valine. These amino acids have completely different electrochemical properties, and that reflects on damage to the quaternary structure of the protein, meaning its shape is no longer going to be the same. And this translates into the sickle phenotype. The sickle phenotype in some regions where there's high malaria, it's a positive trait. It's not such a negative trait in certain regions. Within regions with high malaria, you have a plasmodium whose life cycles involve a gestation period within red blood cells. If the phenotype of the red blood cell is sickle, these cells will probably rupture before the plasmodium reaches full maturity. Therefore, you interrupted the life cycle. Let's look at the life cycle of red blood cells. Usually, you have about 3 million red blood cells being formed every second. And this is the life cycle. They're formed within the red bone marrow, and 90% of them will live 120 days and then its components get recycled by macrophages. 10% of them will just naturally hemolyze and get released in urine. If your urine is of a reddish color, it's because this process is happening in a detrimental manner. But naturally, we remove any red blood cells that have hemolyzed. We recycle amino acids. We recycle the iron, and this gets transported by these globulins. One of them is transferring. It sends it back to the red bone marrow. The heme group, that inorganic moiety, gets converted to bilirubin, and then from bilirubin gets released through the gallbladder into the large intestine. From the large intestine, this bilirubin gets converted to urobilins or stercobilins. Some of those bilirubin intermediates are going to be eliminated in urine, and others eliminated in feces. This is what gives your excretions their particular color. Let's look at red blood cell production. The process is known as erythropoiesis. It occurs as the red bone marrow, and is usually promoted by the presence of erythropoiesis stimulating hormone. I want you to focus on these components: the myeloid stem cells, the proerythroblasts, the erythroblast, the reticulocyte, and the red blood cell. So the reticulocyte is just it's going to mature into a red blood cell, and you have a reticulocyte whenever this erythroblast basically loses its nuclei. Within this transition, you have a high upregulation of protein expression, specifically hemoglobin. And prior to that, you have your proerythroblast. All right, let's talk about blood types. Blood types are going to be determined by antigens. What are antigens? Antigens are the surface proteins or glycoproteins that allow that give a particular identity to a cell, a virus, a bacteria. In this case, we're talking about red blood cells. The particular identity is given by 3 types of antigens: A, B, and D antigen, or the Rh antigen. If you have an A antigen, you will we will have antibodies towards the other antigen, towards the B antibodies against B. If you don't have any antigens, you will form both antibodies. And this makes sense. So if you have both antigens, A and B, you would not make any antibodies against them, because if there are antibodies that recognize the antigens, this is going to happen, a chlorine reaction, agglutination. So this is an example where you have B antigens and you have antibodies that recognize the B antigen. Whenever that happens, antibodies they work on a lock and key mechanism basically, and they're just gonna bind to it and create this agglutinate. This could be detrimental, especially whenever you're thinking about blood transfusions and so forth. But this is also the basis for blood type testing. So if you have an A antigen and you expose it to an antibody that recognizes the antigen, you will see this agglutination process occur. This is also particularly important whenever there is a mother who is Rh negative, who does not have the Rh antigen, and who is pregnant with a fetus that is Rh positive. Usually there is a barrier between maternal blood and the fetal blood, because the mother has a lot of antibodies just circulating in her, and this is one of the cases where that could be a problem. If you have the fetal, especially during the late periods of pregnancy, if there is any sort of contact between maternal blood and fetal blood, this would expose the mother to the Rh antigen for the first time, and her body will produce antibodies against it. Now, this it will be a problem if there is a second pregnancy in which the mother is pregnant with with a baby that has an Rh positive phenotype. Now she has the antibodies against that Rh positive antigen. Those antibodies will bind to the Rh antigen within the fetal blood. There is a treatment called ROGAM in which the mother, during the late stages of pregnancy, she's given an version of the antibody against Rh. This antibody does not cross the blood-brain barrier, the maternal and fetal barrier. But if there is any sort of exposure, what the antibody does, it basically masks the Rh antigen. So even though the Rh antigen and Rh blood might be in contact with the maternal tissue, the mother will not recognize it because it is masked by that ROGAM. Let's talk about white blood cells, aka leukocytes. So that is a synonym for them. These are the role of leukocytes: defend the body, remove toxins, and they do so through phagocytosis. You have here there is we have this, we mentioned this granular leukocytes, neutrophils, eosinophils, and basophils. Neutrophils and neutrophils are going to be your most prevalent. Eosinophils are going to be, both of them will have phagocytic functions. Basophils are involved in an inflammatory response. So they will release histamine and heparin that will recruit this inflammatory response. One thing that I want you to notice here is the shape of the nuclei of these cells. This should be odd. Like usually nuclei are round and they're singular. You don't have these multi-lobe nuclei. This is one nuclei that is just linked by these small stalks. Other cells don't have that. And one of the theories out there is this form reflex function theory, where they are phagocytic, they are highly amoeboid in movement, and maybe a round nuclei is not conducive towards that type of movement. Now here you have the agranular leukocytes, you have your monocytes. Once again, look at that nuclei. It has that amorphous multi-lobe nuclei, and these monocytes, they're also known as macrophages whenever they infiltrate a tissue, because there are infections happening throughout all of your body, and these monocytes need to reach those sites, and they do so. And once they do so, whenever they leave the blood, whenever they leave the circulation, they are known as macrophage. Macro, big, phage, it eats, just like Pac-Man. Now here we have lymphocytes. Lymphocytes, as you can see here, there are three types of lymphocytes. You have your B lymphocyte, T lymphocyte, and natural killer cells. Your B lymphocytes are going to be responsible for the formation of antibodies, and antibodies that if there is a novel virus out there, you would not have antibodies against it. You have your T lymphocytes that are involved in just direct contact with foreign invaders, pathogens, and you have your natural killer cells that are responsible for basically killing cells that have been either infected with virus or that are cancerous. These are just definitions towards these particular maladies: leukopenia, abnormally low white blood cell count; leukocytosis, abnormally high white blood cell count; and leukemia, extremely high number of leukocytes. The difference between these two is that usually leukocytosis is a sign of an infection if you have a lot of white blood cells, and leukemia, you will have a lot of white blood cells progenitors. So the cells that can divide, they are the ones that increase in number, and usually this is a sign of just cancerous phenotype. We talk about white blood cell production. They come from myeloid and stem cells, and they're stimulated by colony stimulating factors. Whenever you can stimulate the formation of white blood cells during chemotherapy, whenever the immune system is compromised, Neupogen is used to stimulate the production of white blood cells. There's a lot going on this graph. What I want you to focus on is on the end result of it and its initial cells. So here we mentioned lymphocytes that come from lymphoid stem cells that come from the hemocytoblast. This is the cell that starts it all. We saw erythrocytes that come from myeloid stem cells that are stimulated by EPO. All other cells, they come from myeloid stem cells as well. All other white blood cells and platelets. Platelets come from these megakaryocytes. All right, let's talk about platelets. This is a view of a neutrophil and platelets. This is a platelet, this is a platelet. This right here is a congregation of platelets. Here's another platelet. As you can see by the 8 micrometer long red blood cell, platelets are small. They circulate throughout the blood, and they come from megakaryocytes, and they are stimulated to be formed by these components. Their functions involve clotting, and once the clot has formed and the whole clotting reaction has occurred, they will contract, basically using actin and myosin, and bring the edges of the rupture together. This process is known as hemostasis. The whole process is known as hemostasis, homeostasis of your blood vessels. What happens whenever, what breaks homeostasis of a blood vessel is rupture within the blood vessel. This process tries to contain that. So we have 3 phases: a vascular phase, a platelet phase, and a coagulation phase. The vascular phase is highly mechanic at the level of the blood vessel itself. You have the smooth muscle within the blood vessel contracting. You also have endothelial cells within the blood vessel just coming together in an effort to close this opening here. So the lumen decreases. We're basically trying to cut off the circulation towards this area until we can control this rupture. The vascular phase involves the aggregation of platelets. Whenever you have a rupture, the collagen, which is part of the wall of the vessel, gets exposed. And that is where platelets attach to. As soon as they see collagen, they're going to attach to that, and they're going to initiate this process of aggregation and release these chemicals that are going to they're going to induce a positive feedback to aggregate more platelets and subsequently initiate these clotting cascades that we're going to see in a moment. They form this platelet plug here. So let's look at this was the vascular phase that we mentioned, and this is the platelet phase. Now let's look at the coagulation phase. The coagulation phase has, the end goal of the coagulation phase is turning this fibrinogen, which is readily flowing in the blood, into these fibrin fibers, this network that is able to seal off the rupture. How do we get to this transition of fibrinogen to insoluble fibrin? Through these 3 pathways: extrinsic, intrinsic, and common pathway. So here we are. Once again, focus on the end results here: the transition of fibrinogen, which is soluble, readily flowing within the blood, to insoluble fibrin that just creates this network that seals off this cut. This fibrinogen, all of these series of events occurs. Thrombin induces this transition, and prothrombinase induces the formation of thrombin, all of which is initiated by factor X. Factor X is the common place where all of these 3 pathways, intrinsic, extrinsic, and common pathway, meet in order to promote the formation of insoluble fibrin. Here we see this fibrin network, these fibrin fibers, starting to seal off this rupture. So at the beginning, fibrin is insoluble. It's just flowing there, protein flowing until this reaction occurs, and then it starts creating these networks, these long fibers that are there to seal that off. Once we have created this clot, the final phases involve clot retraction, in which platelets contract using actin and myosin, and they seal off the puncture. And eventually we need to get rid of this fibrin network that we created. This is done through fibrinolysis. and this helps dissolve existing clots, and this is done through an activated form of plasminogen, which is plasmin. Okay, so that was it. I hope that we have learned about the blood, its different components, its function, white blood cells, different types of white blood cells, um, how the white blood cells are formed, and eventually the clotting reaction to maintain hemostasis. I hope everyone is doing well. I miss you all, and we'll see each other soon, hopefully in person, if not through video. Bye.